Good morning, everyone. My name's Professor Tony Bjorsen, and thank you to the organisers for inviting me to speak to you and outline the objectives of personalised medicine and giving me the opportunity to describe to you how it relates to the COVID-19 pandemic and also to overview how we have responded to the pandemic here at the University of Ulster. Our Northern Ireland Centre for Stratified Medicine is located in Derry, in the Altony Galvin Hospital site, and we work closely with the Faculty of Computing and Engineering and our colleagues in biomedical sciences based down in Coleraine, and indeed with our colleagues in our campuses in Belfast and including the Queen's University of Belfast. And CTRIC is a company wholly owned by the University of Ulster, Derry City Council and the Western Health and Social Care Trust. And our Stratified Medicine Centre was established in 2013 with an investment of 11.5 million. Centre for Stratified Medicine includes 15 academic research staff, about 20 research associates and 25 PhD students and 50 affiliated clinicians, all working in the Western Health and Social Care Trust, and we're all working together on what is referred to as personalised medicine. And we work across a range of diseases as shown here. We also commercialise our research, and we also provide specialist training in personalised medicine through the provision of an undergraduate degree programme and postgraduate courses in personalised medicine. Personalised medicine is largely influenced by our personal genomes, or the genes that we carry, and it aims to, for example, identify an individual's predisposition to specific diseases and potentially enable the prevention of disease onset or delay disease onset. It also aims to accurately diagnose disease, and pharmacogenomics uses our genetic makeup to identify the best treatment for existing disease. And the same can be essentially applied to COVID-19, where we want to ensure the right diagnosis and the right treatment for the right person at the right time. This slide shows the deaths from COVID-19, and you may recall that the first COVID-19 positive cases were detected in Northern Ireland on the 27th of February 2020. And there were 77 more people who tested positive in the 3 weeks running up to the 19th of March. And as a consequence, just before the St. Patrick's Day holiday, we shut down all our clinical research of patients. Basically because these patients were at risk of infection and they were generally vulnerable at-risk patients, and most staff therefore started to work from home along with most of the population. At the outset of the pandemic, many of us in the university sector realised that we had the core skills required to help the NHS fight the pandemic, and therefore we set about setting up a COVID task force to provide RT-PCR testing, antibody testing and contact tracing, working hand in glove with the NHS. We focus on undertaking research that enables the isolation of what are called biomarker panels, and these are based on using genetic data or lifestyle or phenotypic or environmental data to develop tests that can better predict susceptibility to a particular disease or better diagnose a particular disease or determine the stage or evolution of disease with a particular focus on response to treatment. In the early stages of the pandemic, testing for COVID-19 infection in Northern Ireland was initially undertaken only by the Regional Virology Lab at the Belfast City Hospital. So Ulster University and the Western Health and Social Care Trust Altony Galvin scientists formed a partnership to establish manual COVID-19 testing. And that was set up as a manual process that was involved extracting samples manually, and we had no automation at that stage. Getting your hands on and procuring new automated equipment that were required for testing through the NHS proved notoriously slow and bureaucratic, and it takes months, and that was time that we did not have, quite simply. Every developed nation on earth was setting up COVID-19 testing, and there was a massive shortage of the core equipment and reagents and chemicals needed. Initially, we had to resort to making up what are referred to as homebrew test kits from reagents that we could gather up across the university labs, and transferred our equipment from the university labs into the hospital labs to assist with the testing. Within Ulster University, we had already established a COVID-19 task force, and on the 27th of March 2020, the Chief Medical Officer invited Ulster University, AFBI and Queen's to establish a COVID-19 consortium to help coordinate the scarce resources and provide expert testing for the NHS in Northern Ireland. We very quickly realised that the manual testing that we were undertaking wouldn't be sufficient to meet the demand that was coming down the line, and therefore we needed to acquire automatic equipment, which we couldn't procure through the normal NHS processes. And therefore the University of Ulster launched a crowdfunding initiative, and we raised 112,271 in 4 days. And we owe a huge gratitude to the public for their generosity, and that equipment is used to this day, both for clinical diagnosis of COVID, but also for our research and COVRES. On the 16th of June, Robin Swan, the Health Minister, visited Seatrick to see the swabbing facility at Seatrick and also to see the newly installed publicly funded equipment that we provide now in the testing labs at Altnagelvin. Our biomedical science, stratified medicine and computational staff also worked with the Department of Health in contact tracing and developing that technology, and also in the development of COVID-19 tracker that monitored cases and showed figures and movement of people and the number of deaths by day, week and month. The core personalised or stratified medicine questions that arise in the context of COVID-19 would include, for example, why do some people only get very mild symptoms, whereas other people get long COVID, albeit they might have had mild symptoms? Why do some people get really severe symptoms or in worst cases die? Also, why, when we get vaccinated, why do some people react differently to the vaccines with no symptoms whatsoever, whereas others are maybe have mild symptoms where they're laid up for a day, and others maybe have slightly more severe symptoms? Even at these early stages, we know that the answer to some of the previous questions raised lie in our DNA sequence, our genes. And our DNA sequence makes up what we call the human genome. And the genome is a term used for all the DNA that's found in the cells in our bodies. And that's made up of 3.2 billion letters composed of just 4 letters, A, C, G and T. The building blocks, or the instruction manual for how to build and sustain us as humans, or any animal or life form for that matter. And that's roughly equivalent to 200 1,000-page phone books worth of information in each cell. If you think we've got around 100 trillion cells in our bodies, 100 trillion cells can possibly retype new copies or new versions of the 3.2 billion letters every 24 to 48 hours or so. And that's a colossal task. And our cells have the equivalent of spell checkers. They check the spelling of the 3.2 billion letters and they correct the spelling mistakes. And if those spelling mistakes aren't picked up, they give rise to what are referred to as mutations. So, along with environmental exposures and lifestyle, our DNA sequence or marks on our DNA sequence can determine how we live and indeed how we die. It may record if we're very susceptible to COVID-19 or not, and if we experience mild or severe symptoms to infection or in response to vaccination or any other treatment. So that has been one of the important research missions of the Northern Ireland Centre for Stratified Medicine from the outset. And as you all know, we inherit about half of our genome from our mums and half from our dads. And that's a bit like shuffling up a deck of cards, and the genome is equivalent to the deck of cards we've been dealt from our parents. So that shuffling of our genes that we inherit from our mums and our dads, it ensures we each have a very unique DNA sequence. No two of us are identical, apart from identical twins possibly. You can see here you have two individuals with essentially the same series of letters in their DNA in this gene sequence, apart from here at this G position, it's changed to an A in this individual, whereas this T is changed to a C. And these are known as SNPs. And it's these SNPs that can determine if a gene is more active or less active or possibly turned off. And that's what in part can determine whether or not we're susceptible to a particular disease, or indeed susceptible to COVID-19 infection, or susceptible to severe COVID. So identifying the like of these mutations and variations is at the heart of what we do. In addition to that, our lifestyle exposures and stressors to drugs and medication and diet can put other marks in our DNA. It doesn't change the sequence, but it can determine if the gene is turned on or turned off. And we can also read these marks, which are known as epigenetics or methylation on the DNA. At an early stage in 2020, we wanted to answer some of the questions I've shown in the previous slide, along with multiple other international researchers. So we set up what we call the COVRES study, a personalised medicine COVRES study, which is joint with Trinity College in Dublin, and is funded by the Department for Economy in Northern Ireland and the Science Foundation in the Republic of Ireland. And it involves recruiting 1,000 patients COVID patients across the island of Ireland. 50% of the patients will have experienced mild symptoms, whereas the other 50% will have experienced very severe symptoms and been hospitalised, and sadly in some cases may have died. So we're collecting saliva and blood samples from, in fact, there's 518 patients now in Northern Ireland we've recruited and already extracted samples from. And we're collecting lifestyle data, and that might include diet, exercise, smoking, their occupation, looking at their clinical data, if they were hospitalised. And then in partnership with Genuity Science in Dublin, we're undertaking the huge task of reading the genetic sequence, the 3.2 billion letters that makes up the genome of each of those patients. And we're also looking at the methylation marks on the DNA. Those are the marks that are made on our DNA as a consequence of some of our life stressors or exposures to chemicals, or as a consequence of taking medications, or as a consequence of being infected. So all of that data we're recording, and we're subjecting that to artificial intelligence computational analysis to identify the different features in our genes or our clinical data or our medical data that correlates with severe COVID versus mild COVID or response to treatment. That project is probably in the order of 1.2 million to deliver that. We were helped through the work of Genuity Science in Dublin and the whole genome sequencing, and also got significant discounts from Illumina for the methylation analysis. You can imagine as well that that amount of data generates a massive amount of storage requirements, particularly for the genetic data. The raw data on the genome from one person is equivalent to roughly 1 terabyte of information. And I would like to acknowledge Seagate in Derry and Dell for providing us with the core computational infrastructure to store some of that data, along with deposits we'll make in the European Genome Archive. So hopefully that research will put us in a much better position should the pandemic continue and we're faced with challenges from new variants. Hopefully we might be in a position to be able to identify which of us might be more susceptible to COVID-19 variant infections, which of us might be at risk of severe disease, and what treatments might be most suitable for us as individuals should we become infected and hospitalised. The lessons learned and part of the aftermath, there's a great opportunity to realise legacy value through multidisciplinary and the multi-sectorial partnerships that have arisen as a consequence of the COVID-19 pandemic. If we learn from those lessons, if we work much closer together as an academic sector and a healthcare sector and an industrial sector, we'll be very, very much better placed to addressing the challenges of a continuing pandemic or indeed a future pandemic. And in so doing, we'll also be able to generate new ideas, new products that will enrich and increase the wealth in Northern Ireland. In the not too distant future, we're all going to have access to our whole genome sequences of 3.2 billion letters. So the time is not too far away when clinical decisions will be based on predictive evidence from genetic tests, and GPs and clinicians generally and pharmacists will have additional roles as vital information managers, or will require the support of information managers, and they will need a lot of input from multidisciplinary teams. That'll need to be fed through into medical and genomic education, and that background information and understanding of personalised medicine will be vital for our clinical colleagues and for the public in the not too distant future. So thank you very much for listening. I'd like to finish off with acknowledgements to colleagues from the University of Ulster, the Western Health and Social Care Trust, and all our funders and industrial partners and our grant providers. So thank you very much.
My name is David Gibson. I'm a senior lecturer in stratified medicine with a research interest in biomarkers and inflammatory pathways involved in the pathology of arthritis. And in the past year, I've been involved in projects looking at the immunology of COVID-19. So today, I'd like to overview a number of projects on COVID-19 and tie in some of my research as well on arthritis. So I'd just like to give you some background on the centre to start off with, and then also explain some of the similarities between COVID-19 and arthritis pathology. And then I'll give you an overview of some of the projects that have been involved in which are relevant to COVID-19 and highlight the key research questions we are trying to address and explain why these are important and give you an update on any findings that we have to date. So, as I already mentioned, I work in stratified medicine. This is a research centre based in Londonderry at the Citric building on the Altna Gavin Hospital campus. So there are a number of disease areas that are researched at the Centre of Stratified Medicine. I lead the inflammatory disease group, and the primary focus there is rheumatoid arthritis. There are experts who lead in other disease areas, including cardiovascular disease, diabetes, cancer, and mental health. And the common thread between all of these conditions is that they involve inflammation. There's a common feature, and another interesting factor is that many of these conditions occur at the same time in individuals over their lifetime, and as such are called comorbid. So with the expertise that we have in the centre and the skills that we've developed in research since being set up in 2013, we've been able to apply some of this to COVID-19 research. So on the right-hand side of this diagram are many of the projects just below the rheumatoid arthritis block there, which have been going on the last 5 or 6 years. Biomarker discovery, where we look at proteins or DNA to find patterns which tell us how patients would respond to treatments, and being able to give expensive treatments to patients who respond is one of the key aims of that particular study. Moving to the right, I've also been developing a monitoring device which can be used by patients at home, and I'll tell you a little bit more about that. And also I'm interested in developing novel therapeutics and investigating the pathways involved in rheumatoid arthritis. But today I'd like to highlight some of the projects that we've been focusing on in COVID-19, and in particular those that have helped us understand the disease better and investigate what's happening in the Northern Ireland population. Just to give some context about why somebody like myself, who's been involved in arthritis research for some time, would be particularly interested in COVID-19. It would appear that there is much of the pathology is shared between these two conditions. They both involve the immune system and an overreaction, if you like, within the immune system. So there are some similarities and there are some distinctions. So in COVID-19, musculoskeletal symptoms or symptoms of arthritis can develop during coronavirus infections, as with other respiratory infections. There doesn't seem to be any association with how severe COVID-19 is, but roughly 15% of COVID-19 patients would report joint pain, and approximately 44% would report pain in their muscles. COVID-19 is distinct in that it is a viral infection of lung tissue. In severe cases, there is a cytokine storm, that is the early response of the immune system, or the innate response as we call it, develops an overreaction to the virus. And cytokines are small molecules produced by immune cells and the surrounding tissue, and it would appear in severe patients that there's an overproduction of these cytokines, which then goes on to create damage to the tissue affected and can cause scarring. In rheumatoid arthritis, it is autoimmune, so it can take a long time to develop this, and it is a reaction to the body's own tissues and the immune system's overreaction to those, which causes chronic inflammation and production again of cytokines at a lower level, but over a long period of time. And it is the adaptive response, or a different set of the immune cells, which can cause this. And it is the immune system reaction over a period of time which causes, just shown in the top right of this diagram, a cross-section of a joint. On the right-hand side is an arthritic joint, where you can see on the far right of that diagram that the tissues surrounding the joint become inflamed, expand, and can start eating away at the bone and the joint surfaces and cause permanent damage. So both have these features of inflammation, involvement of the immune system. Different components are involved at different times, and it would appear that in COVID-19 there's an explosive short-term overreaction of the early immune responses and production of what we call inflammatory cytokines. There are also immune cell processes which are common. So we talk about different subsets of immune cells, and T cells are one type that are very important, and macrophages are another. And what is common between both is that these two cell types can ingress or invade into the tissue affected and cause scarring in the instance of COVID-19, and destruction of joint tissue in arthritis. With regards to crossover, if you like, between these two different conditions, arthritis drugs which are anti-inflammatory and can dampen down that cytokine storm, are being trialled to treat severe COVID-19. So these are drugs traditionally given to reduce disease activity in arthritis. There seems to be some success with those. And conversely, patients who are treated with drugs which suppress the immune system and suppress disease activity in arthritis can therefore be at risk from an infection and COVID-19. So you will know of many people who've had to shield because of the conditions they have and the treatments they receive. Now, one of the first studies I'd like to tell you about is what we call a seroprevalence study. This type of study is really focused on trying to determine what proportion of the population have had an infection at any point in time. And the latter point is really important because we need to survey throughout the pandemic to see how many people have been infected. So this is a continuing project, and this is a project which has been a collaboration with Queen's University Belfast, the AFBI Agriculture Unit in Belfast, and also the Public Health Authority, along with Ulster University. And the idea with this project was to get a random sample of people who have visited their doctor or been through the hospital system at various stages over the past year and continues into the current year. So we sampled 1,200 people in June of last year and November, looking for antibodies which would tell us they've had previous infection. And we found in June that there was 3.9%, as shown in that diagram below. And in November, that had increased to 5.9%. And you can see at the time points we've taken, they're just after the first wave in the spring of 2020, and then just after the start of what was the second wave and the lockdown in November. It's now estimated that in April, from Office of the National Statistics in the UK, that 60% of adults in Northern Ireland would have tested positive for antibodies. But this is a mixed picture now because many of the population have been vaccinated in Northern Ireland and across the whole of the UK. So they too will produce antibodies. Now, over time, we know these antibodies can disappear and reduce in our circulation. And what's an important question still to address is just what percentage of the population would continue to have protective immunity. And that brings me to the second study I'd like to talk to you about. With this particular project, we've been trying to develop tests which would look at the cell component of our immune system, and specifically T cells, and be able to try and reduce the amount of time it takes with current test formats, make sure it can be done in whole blood, and also be able to differentiate between people who have been infected in the past and also those who have been vaccinated. So with this particular study, it's still ongoing, and we're due to receive results in the next week from this particular study. But just shown in the top right is how we go about this. We can extract immune cells from previously infected individuals from their blood and stimulate those with peptides or small parts of the virus and see if there is a reaction from the cell production of one of these inflammatory cytokines and measure that. And in the lower diagram, we're able to see in the top two set of diagrams the reaction of an individual 4 months after the original infection who had mild disease, that is, didn't go into hospital. And you can see, just comparing the left-hand square to the right, there is a signal just highlighted with the blue dashed line on the right diagram, which indicates that this person is having a T cell specific reaction to these virus peptides. In the lower quadrant as well, which is potentially reassuring, is somebody who is 12 months out from their original infection, had severe disease and were hospitalized. And indeed, again, they are showing a strong response from their T cells. And really, this reflects a memory of the virus, and importantly shows that there continues to be protective immunity in people who've been infected. The types of applications of this technology or tests that we're trying to develop is that it could be used to assess groups who've had to shield with long-term conditions such as arthritis or cancer, and determine whether they need vaccine top-ups. It could even be used in the context of travel, and some level of quality assurance around whether people continue to have immune protection after vaccines or an infection, and could even be used to inform vaccine policy. For example, we have heard all about variants of concern, such as the Delta or variant which originated in India. This was a project which was part of a joint consortium of 7 different universities around the UK, and we were funded by UKRI, and as I say, this project continues. So we're excited to see if we can develop some of these new assays. The third project I'd like to highlight is trying to look at what are the genetic features which are associated with severity. And in a second step, we will hope to look at what is different about the immune system in people who suffer severe disease versus mild disease. Just in the lower diagram, in what we called COVRAZE 1, or stage 1 of this particular study, we have recruited over 500 people in Northern Ireland as part of an effort to get 1,000 individuals in the whole of Ireland. And looking at their genomic sequence, we hope to pick out patterns which would tell us what are the risk factors that people carry, which may explain why they then go on to suffer severe disease and end up being hospitalized. The second stage of this study will aim to look at the immune system and how it differs. Are there specific patterns that would differ in people with severe disease? Do the levels of protection differ from the immune system in people with severe disease? And do they suffer from ongoing health issues such as long COVID? And again, we've put an application in for funding for this second stage, and are hopeful we can continue to follow up the patients that we've recruited. The COVRAZE 1 study was a joint study with partners in Trinity College Dublin, and was jointly funded by the Department for Economy in Northern Ireland and Science Foundation Ireland. A fourth project I'd just like to mention is where we're looking to develop medical devices which could potentially help people at home and extend healthcare services into the home of patients who have had to shield during COVID-19. So we've just recently received funding from First Arthritis, and this is a project which will work in partnership with AstraZeneca and other clinical research organisations, and also partners in the NHS. What we hope to do is to develop and evaluate a prototype device shown below, this blood tracker, which can help to take blood samples from patients at home, and therefore help to monitor the disease activity and afford opportunities potentially to adjust treatments when somebody is at home. And the aim of this project is really to gather ideas from individuals living with arthritis, from laboratory, clinical and industry partners. And this is a 2-year project, and its aim is to bring this device to market. The potential benefits for individuals who could use this device, for patients in particular, would be to provide greater convenience, reduce hospital visits, and really empowers the patient to be in charge of their own condition and part of looking after themselves. For the clinician, data could be available to adjust those treatments, and for a healthcare provider, it could reduce costs and offers that level of COVID-19 resilience. And for clinical research organisations, such as the pharmaceutical industry, it could reduce trial costs by reducing participant dropout. The last project I'd like to tell you about is one that we're currently setting up in the Centre of Stratified Medicine. This will aim to help set up a new testing service to screen asymptomatic healthcare workers and prevent the transmission of the virus through hospitals into the autumn and winter. This will use a new technology called loop-mediated isothermal amplification, or LAMP testing. It's particularly attractive because it's non-invasive compared to the normal nasal pharyngeal swabs, which you may have experienced yourself. It's a simple saliva or spit sample, which can go into a tube. The tube would be sent to our lab and would be a test result could be produced in as little as 25 minutes. It's also attractive because it's inexpensive, and we are in the process of setting up a new lab at Ulster University of 10 staff. Just on the right is a diagram showing that really it's particularly attractive compared to PCR, which is both are very sensitive, but importantly, LAMP will only report a positive result for the period during which a person is infectious, whereas PCR can still produce positive results whenever the virus is no longer replicating or the person is infectious. So there are many people involved in this particular project. My colleague, Dr. Taranjeet Singh Rai, and this is headed up by the Central Department of Health and Social Care in Northern Ireland and in the UK. Just like to acknowledge many of my colleagues in Ulster University and the local health trusts who've been instrumental in setting many of these projects up. I have also mentioned for each of these specific projects, there are too many people involved in each to mention them one by one, but also just to thank the funders of these projects, which include the likes of UKRI, First Arthritis, Department for Economy, and Invest NI and HSC R&D. So thanks for listening, and I'd be happy to take any questions during the discussion. Thank you.
So I'd like to talk to you a bit about COVID and testing and tracing, which my colleagues Professor Bjorsen and Dr. Gibson are covering other aspects of the work from the university, and this is one that myself and my teams were primarily involved in during the pandemic up until now, and indeed in a continuing capacity. So just as an overview, I'd like to talk a little bit first about testing and tracing pre-pandemic and my understanding of what that was. A little bit about what I was doing at the university prior to the pandemic, and then during my involvement with the testing, what my perceptions of the changing of testing workflows were during the crisis, and how the increasing amounts of data that were coming in as well have led to changes in how contact tracing is done, and talk a little bit about testing and tracing now in this new area we're moving into, hopefully the post-vaccination stage. So my involvement initially before the COVID crisis was very limited. My understanding, however, is that like many other people, I would have known a great deal about how testing was done, but mostly it seems to have been done at a fairly low throughput, smaller scale testing, with any larger amounts that needed doing would be sent to, or any specialist work would be sent to the regional virology lab. And this would be typical for most places, and indeed there'd be large virology labs in capital cities and in other major centres in countries across the world. And there's usually a limited need for testing locally, so it's a fairly specialist service. But there was some being done locally. For instance, HPV testing was being done in part at the Western Trust in Northern Ireland. So there was some local testing, but in a very limited capacity. In terms of tracing, I mean, tracing has been done before, of course, for notifiable diseases, so things like E. coli outbreaks or Legionnaires. and the public health, they would have to be notified to the public health agency, and then they would engage local teams, and they would even do contact tracing and try and work out who people have been in contact with, or what the common denominator was for the cluster. And there's an excellent system for reporting called HP Zone in the UK and Northern Ireland, which had a very good annotation system for looking at all of these things. Prior to this, for our own part, we've been working in the Genomic Medicine Research Group, working on genetics and molecular biology, which is my training. Molecular biology being a broader umbrella term, really covering a lot of things that not necessarily our genetics, but we had been working, for instance, on retroviruses. We've been doing a lot of what's called polymerase chain reaction, or PCR, which a lot of people have heard of now in terms of virus testing. And we had a lot of experience in isolating DNA and RNA from human and other material. One of the reasons really that I became involved. But also more recently, we've been moving into epidemiology, genetic epidemiology, and also bioinformatics, which is really a work around computer-based systems for handling large amounts of data that could be generated from various sources. So really any big data set can be handled using computing. And when that is particularly biological data, it tends to be called bioinformatics. And we've been getting a bit more expert in that recently, and we've been doing a lot of collaborative work. Some of this had been already in the epidemiology area. So, for example, one of the large multidisciplinary projects that we were involved in had a strong genetic component, led by myself. And then also we had colleagues in nutrition, in psychology, in stratified medicine, and also in intelligent systems, who are all working on different aspects of the same project. And as I said, bioinformatics, which is one of the really one of the main tools that we are starting to use now, is used for analysis of large volumes of data. And it's usually clinical or biological, but you can also, in fact, look at other types of data as well. And in many cases, you use what's called the R computing environment. It's very short term, just R, the single letter. And it's what's called an open source tool. And really, I guess a good way to compare that would be, it's very like using an app on your phone. So it's sort of like Google Store, so you can download packages or modules which people have already written, and these would work like an app. So R is like your Google Play, and the modules would be like different apps in that. And a lot of these would have been developed by other people, but they would be freely available. And this really is a very powerful tool, and just the same way as a mobile phone has sort of revolutionized modern life. Really, by using different apps, you can couple things. You can use a single platform to do many different things, so you can couple them together into different very powerful analysis pathways. And also, we've been, in fact, developing some of our own software solutions, and one of these is a tool called CandyMeth, which allows people with only very basic genomics to look at data from epidemiological studies without having to write any code, because although we can write code, it's not everybody's cup of tea, and a lot of people in biology or in psychology or in nutrition maybe don't have the time to learn themselves, and this is of course very reasonable, but would still be very interested in looking at the type of data that we have. And these workflows can be developed to allow other people to use it much the same way, as I said, like an app. So you just plug in certain things that you're interested in, and then the software runs in the background and generates the data for you. And even recently, we've been applying some of these to large data sets and becoming interested in looking at a mapping of these particular cases, for instance, or for people who are participating in our epidemiological studies. This is part of the TUDIS study here, and the map was kindly generated by A.D. Moore's group. But this was in collaboration with nutrition and many of our other colleagues there. And so this is the type of application you can look at. It's very powerful. And this is the sort of ongoing work we had when the COVID crisis overtook us. So what did occur during the COVID that sort of engaged our attention? Well, really, this is an interesting slide I found from when I was first reading about COVID-19 back in the start of last year, like many other people. This is a very useful presentation from some colleagues in London Business School, just comparing different pandemics. And I just draw your attention to these more recent pandemics, the initial SARS pandemic in 2002-04, which was SARS-1, or as it's would be called now, and SARS-2, which is COVID, has overtaken us now. And the initial SARS virus caused quite a small number of cases worldwide and a small number of deaths, but a relatively high proportion of the cases. And then things like flu, which of course have caused a great deal more fatalities over time. These are things, both of them can be tested for, and indeed are tested for in local trusts, but testing really never had to reach the type of large scales that we've been looking at recently. I would just note here, of course, that SARS in South Korea, they did do fairly large scale testing for SARS and indeed a related virus called MERS, which is in the same family and is also quite serious. And this is one of the reasons that South Korea was better prepared than many European countries for the SARS-2 or COVID outbreak. So, well, what happened last year, and I think, of course, most people are aware that there was really a crisis that really developed in terms of testing and also in terms of, the system really being overwhelmed with numbers of cases, and testing had to be increased. And by late spring, really, this was never planned to have that type of capacity. And really, regional labs were asked in Northern Ireland to try and develop testing streams as well to help with testing. And this was during this period that the Northern Ireland COVID-19 testing consortium was formed between the two universities and also the Agri-Food and Biosciences Institute, AFBI, and the ALMAC group to try and help where we could. And one of the main problems really with testing was that because of the exponential demand worldwide, really, supplies of reagents that we would normally use in our lab had just become suddenly like gold dust, and you couldn't get them anywhere. And this is because they're the same reagents you would use to isolate RNA from viruses, and also the machinery and the robotics that you would need to isolate samples at a high scale were really being sold faster than they could be made. And a number of these really end-to-end solutions, which were robotic workstations for doing the whole workflow, were built in South Korea, and they had really already developed this because of their experience in earlier pandemics, or epidemics, I should say. Then it's never really reached a pandemic scale. So we were in part tasked with trying to help to source supplies and equipment for alternative workflow. And this is just an example of the type of workflows we were trying to work out at the start of the changeover period there. So there are many stages to these, to the isolation and identification of viral material. So first you have to, if you take a sample from somebody, you have to inactivate that, and that requires one part of the workflow and certain types of solutions. The second part, and that has to be carried out obviously in a safe way in particular types of laboratory. And then once it's been inactivated, then you can move on to isolating RNA from that is the material, the viral material, from the background of human cells. So if you take a swab from the nose and throat, carries a lot of human cells as well, And if you're infected, a small fraction of the total material in there will be viral, and you have to be able to isolate all the material, but also to identify in the third stage in the real-time PCR, or sometimes just PCR, you have to be able to identify the viral material in that sort of sea of human cellular material. And these are sort of interchangeable parts of the workflow. So you might be able to do one part of the workflow with one reagent and the second part with another. And then you might be able to interchange or cross over between these, as you can see here. And with our colleagues, in particular at the Western Trust, where we're heavily involved, we found that there really needed a whole new workflow for many of these things. You had to develop new labs because of the numbers of samples coming in, and had to be sequestered away from the rest of the work being done. You needed specific areas to do the isolation of material from the virus, and maybe a third part of the workflow would be somewhere where you would do the actual PCR. And then, of course, there's the reporting, and the reporting has to be done using specialist computer systems as well. So, luckily during this, the whole crisis, I mean, everybody has been so fantastic, and in the trusts and the workforce there have been really stepped up, and everybody contributed so much, not just in work hours, but outside of work, and staff at the university and many colleagues devoted huge amounts of time to try and get all of this working. And by late summer, really, a robotic prep station had been identified and brought in, and by using different combinations of machines, we're able to increase the speed of testing, and also improve the workflows, and more staff were taken on to shorten the time from the start to the end of the workflow, so that people could get the results in a timely fashion, which of course is so important as well with an infection like COVID. And so really a huge number of tests have been done in the last year through the adoption of new workflows and the scaling up of services and the contributions of new staff. And really, it's a testament to the skill and dedication of the staff there. As well as the one of the things I guess we've learned from this is one of the machines, the robotics that was brought in, is a high throughput device. They also has a low throughput, very hands-off device, which is called GeneXpert, which is there. But also, as the pandemic has progressed, there have been more and more types of testing machine have been developed, and these have now been tailored really, or adopted to different types of purpose in the trust. So you can have ones which are particularly suited for helping with patient through flow, or ones which are good for asymptomatic monitoring of staff. And Tony and David are going to, I think, talk about the LAMP testing, which is this non-PCR based method, which is excellent for rapid testing as well. So there are of course similar initiatives at trusts across Northern Ireland. So for the next part, I'd just like to talk a little bit about contact tracing, which is a bit of a changing gear. So rather than looking at things in the lab, this is really mostly computer based. And this was done in partnership with the Public Health Agency. Initially, I came in contact with them through the consortium, and it became engaged there during the pilot contact tracing program, which in fact was the first one in the UK to get off the ground. And it taught us a lot about what sorts of things were going to need to be adapted and put in place before the full-scale contact tracing program started. And it was clear from even the pilot program that new approaches would be needed because of the high volumes of data being developed and the new systems that were being used to collect it. And really, prior to COVID, I think there was a number of different systems around, like HP Zone, which I mentioned, Public Health England were using. In the Republic of Ireland, they didn't have an equivalent, but they adopted a Microsoft solution called Dynamics, which was used then to build a contact tracing system. There was a system called SORMAS in Germany, and in other countries you have other systems as well, and many of them sort of bespoke solutions. And this is from an article published in 2020, looking at the key desirable features for contact tracing, and they identified a number of different softwares that were available already. These are a few that I've mentioned, and then they scored them on different criteria. You'll note here one of them is what's called open source. And in the end, in fact, and we ourselves thought that Epinfo would be an excellent solution, but in the end, in fact, none of these pre-existing solutions had the capacity needed or the flexibility and the security aspects that were needed to deal with the massive volumes of data that were coming in. And I think worldwide, really, there has been a convergence on more or less similar platforms for contact tracing analysis. So this is where you're looking at large amounts of data coming in about who people have been in contact, who has the virus, and then who they might have been in contact with, and trying to manage all of that data in terms of where they might have been and where clusters and outbreaks might be occurring. And none of these off-the-shelf solutions, as I mentioned, were really useful. They were really more suited to smaller scale outbreaks. And a lot of the software is protected as well, and you can't really change it without consulting, and that makes things very slow. As we know now, viral waves come and go over a period of months, if not weeks. And so really speed is of the essence when you're trying to adapt and move with the virus and try and get ahead of the curve, quite literally. So in many cases, most countries seem to have, I think, zeroed in on many of the same types of solution. That is what we call an open source software. That is, it's not really IP protected, and it's not held by any one company. And these can be where you can modify them yourself, but that they're on secure platforms. And it is quite similar to something like Google Play, where you have developers can develop software solutions in a secure environment, but that they're suitable for dissemination as well and adoption. So this is an example really of one of the types of software solutions. I mean, this is just a freeze from one of the pages for one that was developed here in at UU with the PHA for looking at COVID-19 cases. This obviously gives you an idea of where clusters of cases are occurring in Northern Ireland, and it takes advantage of some existing solutions, and it uses this R computing environment that I mentioned, and you can take existing solutions for mapping and for linking cases and contacts, graphing, generating tables and statistics, and use these to help you to get an idea of where clusters are occurring and where you might have linkages between them as well. And it's can take advantage of existing secure systems that are already in place, such as Microsoft Business Solutions, that are widely available to people as well. And really, in a lot of places, what they're doing is developing them in two layers, much like our example of CandyMeth earlier. We have a developer layer, and then we have a layer for people who just want to use it to get information, and that's sort of the user layer. And these solutions can be developed in-house, and they can help you to, for instance, look at clusters, new outbreaks, break down the data by different criteria, like local government district, ward, postcode, in fact, anything for which you can glean data from the public domain, or indeed from the data that is coming in through your protected trust systems. And these can help the cluster management team to determine where active clusters are and which clusters are no longer active, and where they have become under control. And can help you to get an idea of where, for instance, at a workplace, where the cases are spread around that workplace, the workplace here in green, and then the cases where they have where they are then located themselves, and where you might be getting a spread. You can see these people all live locally to their workplace, and And you can get an idea. Many different types of analysis are possible here, and this can be changed and modified very quickly in a matter of hours or days in order to get new types of data and new types of analysis done. So here would be, now this is a bit more complicated looking. This is just, it looks like a series of sort of a short pattern here, but it's what we call a social network analysis. And this is a way of representing connections between cases in this picture. And this was built using by the team based on an existing app, if you like, but has been adapted to fit the contact tracing data in Northern Ireland. And what we have here is we have each blue dot represents a case, and then around these you have secondary cases, which are in red, and then we have, these are people who have gotten the virus from the initial case, and then in yellow we would have people who are their contacts and may or may not go on to develop become infected. And these are all of the cases and all of the contacts associated with one particular fairly large outbreak in a factory. And doesn't look maybe easy to interpret initially, but once you have looked at a few of them, you can pick out different types of transmission. So here I've coded them, for instance, HH for household transmission and H2H for from house to house transmission. This is where a virus would have, so we know, of course, that SARS spreads very easily within a household. So in many cases, you get a number of members of the house who are coming down with it at once. But also you can get transmission from one house to another, and these are particularly important to identify. And here, for instance, in this example, you can see the initial case in blue at the center of the top circle had quite a large number of contacts here. Some of them were living at a second address, and a couple of these, in fact, 3 of them became cases themselves later on, and then they had further contacts. You can see here already a spread from here to a second house, and then onward from there to other contacts. So what has all of this work taught us? Well, I mean, I think in terms of testing for viral infection, I think it's really brought home the point to us that we need to build capacity across the region for testing. And we need to also work together with our suppliers and also, I think, in academic clinical collaborations to help to test new platforms and see what they are most appropriate for. I think it's really important to diversify supply chains to avoid having a single point of failure, so that in case one supplier just can no longer provide you with what you need, you have an independent way of carrying out the test. And one of the things that has come out of this moving forward is the setting up of a hybrid lab between the Western Trust and the university, which is, one of the focuses there will be on testing going forward as well. And it's also really important, I think, to build local capacity and local expertise in these things, so that when and if there is another crisis, that we will be in a position to help and to move more rapidly as well. In terms of tracing, really there's an ongoing need for data analytics, which is really the analysis of large amounts of data using computers as part of health surveillance and outbreak management. And while some was already being done, I think this new component in terms of looking at infectious disease really needed almost a separate team there. And you need flexibility in terms of your computing to allow a rapid movement or rapid adaptation to new circumstances. As I said, the virus moves very rapidly and a wave occurs very quickly. And you need obviously secure data systems and platforms to carry this out. And we're continuing to work with the Public Health Agency to help build capacity there for these things. So I'd like to acknowledge just some of the teams that were involved here, and really this is not exhaustive, but I would really like to thank my colleague Tony, of course, who has been instrumental in leading the COVID response at the university, and Lee McGuire, who was very kindly seconded Ben Wingfield and helped us with contact tracing, and Sarah Jane Thursby, who Catherine have done a lot of work on that, as well as Andrew and a number of members of my team who've been helping with the various parts of the COVID response at the Western Trust. Michael McKenna, Melissa Kane, and of course Mary McMenamin and Marty Robinson, as well as a number of other members of the team there at the PHA, Damien Bennett, and Jackie Highland was involved in the pilot, Hugo, and a number of colleagues there who've been working diligently on data analytics. And I'd like to thank lastly my colleagues who at the university whose data that I showed there a little earlier. And with that, I'd like to thank you for your attention, and I'd be very happy to take questions in the post-presentation phase.